Welcome to Clinical Histology. I'm Dr. Xie from SUMC. We will talk about cardiac muscle in this session. By the end of this session, you will be able to describe the characteristic features of cardiac muscles, and you will also be able to explain the structures and functions of the highly specialized cell-to-cell junctions in the heart, the intercalated discs. Cardiac muscle is an involuntary and striated muscle that is found only in the heart. Highly coordinated contractions of cardiac muscles allow your heart to pump and keep blood circulating throughout the body. On your left is H&E staining of cardiac muscle, cut longitudinally, and on the other side is also a cardiac muscle, but in cross-section. As we see in both sections, as indicated by the yellow arrowheads, each cardiac muscle cell possesses a single, large, round and centrally placed nucleus. In some instances, you may find two nuclei in a cardiac muscle cell. You may recall from our previous session, the skeletal muscle cell has many peripheral nuclei, which is different from the cardiac muscle. Unlike skeletal muscles, cardiac muscles are extensively branched, as evident in the longitudinal section. If you look closely at the same section on your left, you could also find striations across the cardiac muscle fibers. This is similar to that of skeletal muscle fibers. I hope that you have noticed that thin, typically dark staining lines in the longitudinal section, indicated by the green arrowhead. They are highly specialized cellular end-to-end junctions, known as intercalated discs. The intercalated discs run perpendicularly to the direction of muscle fibers and represent the interface between adjacent cells. Under electron microscope, the intercalated disc appears more complex. At low magnification, as indicated by the yellow arrowheads on the left, the intercalated disc is a convoluted electron-dense structure. At high magnification, the intercalated disc appear more irregular and step-like, with both vertical and parallel regions to the direction of the muscle fiber, as shown in the diagram on the far right. Vertical regions of these irregular step-like discs contain many desmosomes and fascial adherens junctions, which together provide strong intercellular adhesion, so the cells do not pull apart during fiber contraction. The parallel regions of each intercalated disc run in the same direction to the myofibrils and are rich in gap junctions, which allow action potentials to spread between cardiac cells by permitting cations to flow from one cardiac muscle cell to the next. These regions serve as electrical synapses, facilitating rapid impulse conduction and coordinated contraction of the entire heart. Let's move on and take a look at other organelles and features of cardiac muscles. As we mentioned above, similar to that of skeletal muscle, cardiac muscle is also striated, and the thin and thick filaments are organized into sarcomeres as well. You can easily find out the dark-colored A-band and the light-colored I-band in alternating pattern. This pattern is the same as that of skeletal muscle because of the similar organization of thin and thick myofilaments in the sarcomere. In this electron micrograph, the Z-discs are indicated by the red arrowheads. As you may recall, the region of the myofibril between two successive Z-discs is sarcomere. Now, I would like to draw your attention to the yellow arrowheads in the micrograph. Please note that the intercalated discs are overlying the Z-discs, at the interface of adjacent cells. Cardiomyocytes also contain T-tubules. However, in cardiac muscle fibers, the sarcoplasmic reticulum is less well organized compared to skeletal muscle fibers. Therefore, a single T-tubule usually only combines with one terminal cisternae of the sarcoplasmic reticulum to form dias rather than trias, which are found in skeletal muscle fibers. Mitochondria are abundant in cardiac muscles. They occupy up to 40% of the cell volume, higher than that of skeletal muscles. The plentiful mitochondria provides a large amount of energy to cardiac muscle cells, fulfilling the energy demands of muscle contraction. In general, cardiac muscle cell contracts in similar way to that of skeletal muscles, although with some important differences, such as the properties of action potential. The cardiac action potential is long in duration and complex in mechanism. For the detailed mechanisms of cardiac contraction, I refer you to the physiology books and our online clinical physiology sessions. The structural features we discussed above endow the cardiac muscles with their distinctive properties of contraction. Finally, I'd like to emphasize an endocrine function of the atrial muscle cells. These cardiac muscle cells house secretory granules containing a peptide hormone, atrial natriuretic peptide, ANP, that functions to lower blood pressure by acting on target cells in the kidney. Diseases which compromise the pumping ability of the heart muscles are called cardiomyopathies. Cardiac hypertrophy is one of the most common types of cardiomyopathies. In cardiac hypertrophy, the number of cardiac muscle cells do not change. However, they increase their size and modify their extracellular matrix, resulting in abnormal enlargement or thickening of cardiac muscle fibers. On your left is a longitudinal section of normal cardiac muscles from a young accident victim. On your right, we have the cardiac muscle from a patient with hypertrophic heart failure, showing enlarged nuclei, longer and thicker muscle fibers. Please note that both slides are at the same magnification. At present, there is no cure for cardiac hypertrophy. However, we can use medications early to control underlying causes such as hypertension and treat symptoms once they develop. Now, let's have a brief review of today's session. Generally, we have dealt with the key structures and functions of cardiac muscles. Cardiac muscles possess some distinct features, including branched muscle fibers, centrally located nucleus, and dials formed by the T-tubule and the terminal cisterna from only one side of its neighboring sarcoplasmic reticulum. The organization and constitution of sarcomere are similar in both kinds of striated muscles, the skeletal muscle and cardiac muscles. Another unique characteristic of cardiac muscle is the presence of intercalated discs. Can you identify the intercalated discs in both light and electron microscope? Can you describe the functions of desmosomes and gap junctions in the intercalated discs? That's all for today. Thank you for joining us. See you next time.